The final lesson in this topic is Lesson 5, Genetic Technologies, which begins on page 48 in your textbook. So, before we start in on the lesson, I want to take a moment to think about something. With genetic modification, which of these items do you think can be or have been genetically modified? Choose all that apply. So A, apples, B, corn, C, salmon, or D, salt. I want you to come to class tomorrow with your response from this. Some of our guiding questions, some of the things that we should learn throughout this section is how do humans use artificial selection to produce organisms with desired traits? How do scientists engineer new genes? And how can genetic information be used? On page 48 in your textbook, there's a connected that reads, dogs come in many different shapes, sizes, and colors. Which of the ones shown here would you prefer as a pet? Circle your choice. So, we have a lot of different options. Now we're going to move on and answer our questions from applying concepts. Many purebred dogs have problems later in life, such as joint or eye diseases. Why are purebred dogs more likely to develop problems later in life? So if we think about this based on the information that we already have and the gizmo that we did in class, what we can assume is that there's not enough genetic diversity within the breed, which causes the problems to be passed on to the offspring within the breed. Ultimately meaning that there aren't enough parents. All of them are, it's becoming a point that all of them are related, and therefore, like we talked about the royal family and genetic disorders, where they pass them down because they intermarried and there wasn't enough genetic diversity, it's the same situation here. Moving on, what can be done to decrease the likelihood of these problems appearing? So the big thing that we can do is mate two dogs from different breeds. This creates a greater genetic diversity and will most likely produce a healthier puppy because we don't have the issues from both sides of the family. So if they have a German Shepherd, which are prone to hip dysplasia and several other conditions, and they mated a German Shepherd with a dog that is less likely to have hip dysplasia, which would typically be a smaller dog, maybe there would be a greater likelihood that you wouldn't have a dog that's going to have hip dysplasia or whatever other negative disorders that come with specific breeds. Artificial selection and natural selection. We started working on this with our gizmo that we did in class, but let's dig a little bit deeper into what all of these things mean. Scientists and breeders influence the traits that organisms inherit. Natural selection, which is what happens in nature, individuals with beneficial traits are more likely to survive and reproduce than individuals without those traits. Like our insects, the green insects were able to survive more easily than insects of different colors because they could blend in. Artificial selection, also known as selective breeding, this is where humans breed only organisms with desired traits to produce the next generation. So some examples that we do this in is cows, chickens, and pigs. In order to get larger selections of poultry and meat, we breed them so that they produce more. So when it comes to the chickens, we want chickens that have larger breasts, larger thighs, larger, all of those parts that we eat on the chicken. We want those to be bigger so that we can maximize how much food we have. With cows, we want the ones that can produce the most milk. So we made the ones that produce more milk. So that way we can hopefully get a cow that produces the maximum amount of milk as possible. And that's the goal. The goal of artificial selection and selective breeding is that you get all of the traits and desires that you want. So what's animal husbandry? That is the breeding and caring for farm animals that have desirable genetic traits. Animal husbandry is also used places like zoos where they're trying to increase or repopulate certain species that are doing not so well in the world because of changes. But most often, farm animals are where you see it. So, how could humans selectively breed a miniature poodle from a larger poodle? How did they do that? Well, when you take your poodles, if you have one that is smaller than the rest, you're going to breed that with another one that's smaller. And then you're going to take which ones of those are smaller and find another dog that is also smaller until you have a poodle that's the size that you want it. Genetic engineering. So genetic engineering is when scientists transfer a desired gene from the DNA of one organism into another organism. A really big example of this, and something that they've done a lot, is using the jellyfish gene for fluorescence and inserting it into fish eggs to produce glowing fish. They've done that to several different species so that they grow in different organisms. Another example, the gene for human insulin was inserted into a bacteria, and the bacteria now produces human insulin that is given to those who are diabetic to control blood sugar levels. Because bacteria reproduce quickly, large amounts of human insulin can be made in a short time. So that's a beneficial genetic engineering that was done that helps us as humans. And the majority of genetic engineering has done just that. Whereas having a fluorescent fish is more of a desired trait, something that we want them to do. Some of the genetic engineering we do that has been done is we do because we need to have something else. So like the insulin, they figured out that if they did put it in the bacteria and the bacteria reproduced rapidly, they could get more than they could from just creating it or getting it in more natural ways. So now you might be asking, okay, how did they use the bacteria to make insulin? That doesn't even make sense. Well, we're going to look at page 51 in your book, figure 3, and we're going to talk about bacteria that makes insulin. So first we have our bacterium, and then they have small rings of DNA or plasmids that are found in some bacteria cells. So the one that we're looking for will have that. Scientists then remove the plasmid and cut it open with an enzyme. They then insert an insulin gene that has been removed from human DNA. The human insulin gene attaches to the open end of the plasmid to form a closed ring. Some bacteria cells take up the plasmids that have the insulin gene. When the cells reproduce, the new cells contain copies of the engineered plasmid. The foreign gene directs the cells to produce human insulin. So, if we look over here at our bacterium, when it goes down, it shows the section of our plasmid ring being broken off. We take the human DNA and the insulin gene, separate it, and insert it into the section that we have removed from the plasmid ring. We then reinsert that back into the bacteria, and then the bacterium is going to reproduce. When it reproduces, it's going to create 2 identical daughter cells, and they're all going to look exactly the same because of mitosis. Okay, so if you could genetically engineer a trait, what would you do? I want you to think of a new trait. So you're going to create a trait that has never been seen before in an animal. Identify a trait you would like an animal to have. Then sketch the animal and describe a process by which you could achieve your desired result. I want you to go ahead and do this in your textbook. which should be on page 50. There's the planet section, and I want you guys to fill that out. It can be anything as long as it's something that could realistically be achieved through genetic engineering. Gene therapy in humans. So what is gene therapy? It's the changing of a gene to treat a medical disease or disorder. So a normal working gene replaces an absent or faulty gene. An example would be engineering immune system cells, or T cells, and injecting them into a person's body to attack and destroy cancer cells. Then there's the CRISPR gene editing tool, which can cut out the DNA sequence, causing the dangerous mutation while pasting a copy of the normal sequence into the DNA. So that would be, say we have a trait for color blindness, right? We know that that's an X-linked trait. It's just in the X. With CRISPR, we could, in theory, take that gene, cut that gene out, and replace it with a working gene. That's the idea behind CRISPR. And yes, it does work. However, I want to note that these therapies are still being developed, and they only help treat a genetic disorder. They can't cure the disease. So it's only able to do that in some of the cells, and it doesn't work for everything. Cloning organisms. So what's a clone? It's an organism that has the same genes as the organism from which it was produced. So here are the steps to create a clone. 1, we're going to remove an unfertilized egg. 2, we're going to replace the nucleus in the egg with the nucleus from a body cell. 3, we're going to place the egg inside a female so it can develop. And 4, the offspring born with the same DNA as the body cell donor. So we'll have an offspring that has all of the DNA of the body cells of whatever the donor is. All right, so some cloning examples. So a lot of plants are cloned through cuttings, and an example is the Cavendish bananas, which were cloned in order to have more for eating purposes. So cloning helps to produce crops of consistent quality. However, with that, you're going to have a lack of genetic diversity. Another example is Dolly the sheep, and Dolly was the first cloned mammal, cloned in 1996. So now we have a question. We're going to theorize that a fungus that causes bananas to rot is spreading across the globe. Why is a disease more damaging to clone crops? Our answer to this is very similar to our answer about the diseases in the dogs earlier in the lesson. And it all stems from the lack of genetic diversity. So if the clone is susceptible to the fungus that rots them, then all of them are going to be susceptible to it. However, with that, we could, in theory, use genetic modification to figure out what is resistant to the fungus and use that to genetically modify the bananas so that we have a clone that is also resistant to that. So now we're going to talk about some practical uses for DNA. First of all, sequencing the human genome. DNA sequencing is when scientists determine the exact sequence of nitrogen bases in an organism's DNA. Your genome is a complete set of genetic information that an organism carries in its DNA. So the Human Genome Project, which was completed in 2003, identified the DNA sequence of the entire human genome. It allows scientists to better understand certain diseases. It shows us genes that we share with other organisms. There are some genes that exist in almost every organism in Earth. Another practical use is DNA technologies. So what are some uses for DNA technologies? DNA screening. It helps diagnose genetic diseases, DNA fingerprinting, which can identify a person who committed a crime, prevent the wrong person from going to jail, identify remains, and identify the father of a child. Which suspect was at the crime scene? We're going to look at the image next, and we're going to see which one has the same gene pattern. And according to this, we can say that suspect 1 is definitely not it because there does not match. Suspect 3 also does not have, which means that our suspect has to be suspect 2. Why can DNA fingerprinting be used to identify a person? So fingerprinting, everybody has their own unique fingerprint. And that is why your DNA fingerprinting can be used to identify a person, because every single human has a unique fingerprint. Ethical concerns and controversies in genetics. What is ethics? So ethics is going to be if something is right or wrong, if it's appropriate, if something is moral. So is it ethical to say take CRISPR and use that to genetically modify babies to get the traits that we want them to have before they're even born? So now we have, what are some concerns about genetic information? There are all kinds that we have. Some people fear that because of having genetic disorders, that they will be, because we know so much that people that have things like genetic disorders, that they will be discriminated against, especially in regards to insurance, medical facilities, even things like jobs and working, that if they apply somewhere and there is a job and they find out that they have this disorder, that they won't keep them. And so that also ties into what are some negative ways that genetic information could be used. So that's one there. What are some concerns about the genetic technologies we have discussed? So like I said prior with the CRISPR, that's a concern. Is that ethical? Can we do that? Is it acceptable for us to change our DNA so that we have the traits that we want, as opposed to something more like getting plastic surgery? You have that, you can do that. The person decides that that's what they want. Now, in comparison, doing CRISPR to have a child that has traits that you want, can they have a say in that? No, the child can't have a say in that. You are deciding what you want. So is that ethical? Controversies of DNA use. GMOs, or genetically modified organisms, are things that change the DNA so the desired traits are expressed. So growing food from seeds that have been genetically modified. The cons, unknown impact on human health and the environment. The pros are that farmers yield more food with GMO crops to feed a growing human population. Genetic Information Non-Discrimination Act, or the GINA law, is a 2008 law that makes it illegal for health insurance companies and employers to discriminate against individuals based on their genetic information.